Tripta, and today I'll be continuing with the experimental sessions which we have been having by introducing you to red blood cells, and we'll try to do some experiments with them. So previously in the last two experimental sessions we have generated GUVs using PVA method, and we have tried to make GPMVs in the previous session, where we had taken hex cells and we had tried to remove all the systems inside the cell, and we made GPMVs out of it. Today we are not going to prepare anything because thanks to nature is preparing the red blood cells by itself. So we are just going to use them and try to understand how they react to the external environment. That is one small bit of an experiment which I would like to make you all guys do with the red blood cell. So first, before we speak about anything else, I need to go to the next slide. So when we talk about red blood cell, we know that it's one of the most integral part of our human body because that lets us breathe. So we know that's very important for our survival, and red blood cell helps us in doing that. Now when we speak about how it actually does that, we know red blood cells actually travel through our entire body, and they contain hemoglobin, which holds on to the oxygen molecules, and oxygen is carried through the red blood cell, through the bloodstream, throughout our body, and red blood cell plays a major role not only in actually transferring oxygen throughout our body. It helps in drug delivery, it helps in maintaining so many aspects and processes in our body, which keeps a basic functioning of our human body intact. Okay, I shouldn't be saying human body because every single animal which has blood in it is surviving because it has blood in it. So when we speak about the generation of red blood cells, which we need to understand to actually how the red blood cell is formed and how the red blood cell is going to function for us to actually know its importance. Now when we speak about the basic functionality of red blood cell, like I said, we not usually speak about the oxygen supply in our body, but not only it does that, it removes the CO2 from our body, and then it actually maintains acid-base balance by controlling the CO2 content in our blood, and it even interacts with the immune cells and help the body to adapt at high altitudes. Actually, red blood cell studies are even done for different people of different who stay in different altitudes, and there are very interesting bit of work which are happening in that area, which I am not going to get into, which is out of scope of the experimental session today. So the generation of red blood cells, the process is understood as erythropoiesis, where the red blood cell is actually generated from hemopoietic stem cell, which is generated in the bone marrow. Now we should know that red blood cells are anucleated in general, but when they are formed initially they are nucleated. Now why, if other animals like birds are having nucleus in their red blood cell, why human beings don't have a nucleus in their red blood cell? That's because we have more requirement as the body functionality. In terms of the functionality, we have more requirement of oxygen, and to hold on to that amount of oxygen which we require for the normal functionality, we are actually removing the nucleus from the actual homeopoietic stem cell, because we are not doing that man-made, it's naturally happening. Our body is so efficient that it's removing in itself so that the surface area of the cell is increasing. And what is that doing? It is able to help us hold on to more number of oxygen molecules, through which there is uniform circulation of the oxygen throughout our body. Now this is the general process in which the nucleus inside the red blood cell initially, which is present inside the stem cell through which the red blood cells are formed, it's gradually removed through a protein called erythropoietin. Now this is actually going to differentiate, and it is going to help us in mapping out the cells and segregating them in order to actually get them into the process of enucleation and turn them into red blood cells. Now we know that red blood cells are enucleated. By that we know that there is no inherent energy generation, so they have to die. Now their life cycle is for around, on an average, 120 days, and after that average period of time where they actually circulate throughout our body and they go through deformation, they go through bending, like Sharpari ma'am and Thomas sir have already mentioned in their lectures, there is a lot of mechanical variations which are taking place. There will be wear and tear, and which they cannot repair by themselves because they don't have that in them. So what happens is that after a gradual wear and tear which they experience due to the circulation throughout our body, there will be a time when these cells will actually die. Now, what do we mean by death of a red blood cell? Now, after their functionality's efficacy is reduced, what happens is that macrophages in the spleen, liver, what they are going to do is they are going to segregate these red blood cells depending on the wear and tear which they have gone through, and they are going to be removed from the body through different processes. And the most we always underestimate our body and we take it for granted, but it is so efficient that even the red blood cells which are dead are actually recycled in our body. We as human beings don't do recycling in our life, but our body does recycling for even the red blood cell, and it doesn't leave even the smallest possible protein, and it recycles each and every aspect. Like when the red blood cell is dead, it will actually be broken into several parts where there is globin in the red blood cell disintegration, which is actually again further segregated into amino acids, and they are again recycled. There is actually bilirubin content which is actually recycled through the body by sending it to the liver, and it goes back to the stream. And then we have iron, the heme content which is there in our blood. That iron content is also recycled in our body, and it again goes to the bone marrow where we are generating new red blood cells. So why should we study red blood cells under microscope? Now, understanding red blood cell morphology and functionality plays a major role. Like I mentioned, they are going through a lot of deformability, different kinds of deformability in our body. So understanding their morphology, mechanical properties, structure and integrity plays a major role in understanding how they are going to function under different conditions and how that is going to affect the function of our body. Now, another advantage of studying red blood cell in microscope is we can actually understand in real time how the red blood cell behavior is going to change. And another most significant aspect where we actually study red blood cell using microscope is that we can diagnose different blood disorders like anemia. You can actually see that these are normal red blood cell. This is an image of a normal red blood cell. Now when we actually look at the anemic cells, you can see that just by looking at the cells, you can see that the morphology and the shape changes are very significant when you look at the anemic cells. So just by looking at a patient with anemia, extracted blood from that patient, we can actually find out that this person is having a particular disorder, blood disorder. Now, extensive study has happened in that area, but I am not getting into that. Another example is sickle cell disease, and in sickle cell disease you can actually see that instead of a discocyte shape which we usually see for red blood cell, they are biconcave in shape. They are discs with a biconcave structure to them. But when a person is actually having the sickle cell disease, their red blood cells are actually like these, forming these shapes, the crescent shapes which you can see here. And this is for malaria, and you can actually see that the cells which are actually affected by the malarial parasite is actually seen in the form of a ring in the center. So just by looking or observing the red blood cell under a microscope, and even the minute observations can actually give a huge interpretation in terms of how the morphology, structure and their functionality is changing due to interaction of them with different pathological conditions, chemical conditions, environmental conditions, physical conditions, like we spoke about stretching, bending through tweezer, AFM and all that. So depending on the force responses and depending on how they are observed under a microscope, a lot of significant outputs and insights can be extracted about the red blood cell. So there are different ways in different types of microscopy which we use to understand red blood cell or cells in general. One is bright field microscopy. In bright field microscopy, it is very straightforward, and Suchita here gave a talk on different kinds of microscopy in her talk. The second very common thing is a phase contrast microscopy, and there are SEM images, AFM images, light sheet microscopy, fluorescence, confocal, and they are all different ways in which you are perceiving the red blood cell. Now what you want to extract out of the cell, the information which you are trying to extract out of the cell, depends on which kind of microscopy you are going to use. And there are many more methods in which you can perceive the red blood cells. So now we understood there are different ways in which we can look at them. Now to look at them, we have to prepare the sample. It's not like we can just extract the blood and put it in the microscope and look at it. So there are different ways in which red blood cell sample is prepared. The first method and most used method to understand the dynamics or to understand how inherently the red blood cell properties are maintained is using a method called wet mount preparation. Now in wet mount preparation, what we are doing is we are observing red blood cell in their natural state. We are maintaining the osmolarity, isotonicity and all the other properties and retaining the properties which are there inside our body. I mean, we are just trying to manage it for a short amount of time, and we are able to see how they are going to respond, like under force, under the influence of force, or if we are actually introducing it to some pathogen, or there are different kind of studies which have happened. Second thing is the blood smear technique, which is a standard technique which even in the pathological labs we actually see, where we are just looking at the morphology, change in the morphology and the structure of the red blood cell. Third thing is fixation and staining. Now fixation and staining is just to enhance contrast and look at the abnormalities, which actually I showed you when I was showing you the images of red blood cell for sickle cell anemia and all that. And the fourth method in which we can prepare the sample is called as an immobilization technique. Now immobilization technique is used for microscopies like AFM, SEM and TEM, where you know that when you are actually looking at red blood cell through the eyes of atomic force microscope, SEM or TEM, we are not looking it through optics. We are having a direct contact. Now when we are having a direct contact with the red blood cell, we know that a red blood cell is actually, most of it is fluid inside. Now if we are putting it on a glass slide and just smearing it, its properties are going to change, and the forces which are applied through AFM are going to actually move them around, and we don't want that because we are trying to understand at a nanoscale what is the morphological change using AFM. So what we do is we immobilize the cell and then we treat it with different chemicals to do that, and then we try to image and understand the force spectroscopy using AFM. So procedure to make this red blood cell for which we are, using which we are doing the experiment today is the wet sample method, because we are doing phase contrast microscopy today for our experiment. Now usually we know that whenever we take the whole blood, it doesn't have only red blood cells. It has white blood cells, it has platelets, it has plasma, it has serum, it has all the other things in it. Now when we take the whole blood, red blood cell is 45% of the whole blood, which is like if you have 100% whole blood with you, only 45% of it is red blood cells. Remaining is the white blood cells and all the other constituents which I was talking about. Now if you want only the red blood cells, we have to follow this protocol and we have to remove the rest of them. What are we going to do is we are going to take the whole blood, we are going to centrifuge it at 100 rpm for 5 to 10 minutes, and then what happens is all the serum, plasma are segregated and red blood cells are going to form pellets in the bottom. Now the supernatant is removed after centrifugation, and these red blood cells are given any kind of buffer with osmolarity 300 milliosmolar per liter or pH of 7.4, and what there are like HEPAS we can use, PBS, phosphate buffer saline, and there are other normal saline. There are different buffers which we can use to actually dilute the red blood cell. Now we'll do that, and what happens is that along with all these serum, platelets, white blood cells, we'll usually have contamination. A few red blood cells are dead using the centrifugation. So what we do is we remove all of them by cleaning or washing the red blood cells, and that we do again by centrifugation, but inside the buffer with which we are going to maintain the conditions for the red blood cell. And that we are going to do by washing the red blood cell pellets which we took. We are going to take it in a buffer depending on the number of red blood cell ratio which we want in that sample, which is called as a hematocrit level. We are going to actually dilute the red blood cell using the buffer, and we are going to wash it. And finally, after 2 to 3 times of washing, we will usually get it to 5 to 10% of hematocrit level. And we do this because for phase contrast microscopy, it is ideal to have so many number of cells in our sample.
How do you measure that hematocrit level?
Now I was actually telling you about how many red blood cells are there when you take whole blood, right? 45% red blood cells are there. Now depending on how many red blood cells you need in the sample you take, like let us say you are diluting it using the buffer. Now when you are diluting using the buffer, you cannot put how much of a buffer you want. You cannot take 1 drop and take 10 microliter or 1 liter and all that. So there are particular reasons why we do that, right? We are trying to maintain the osmolarity and isotonicity, and that can happen only if you have a particular ratio, first thing. Second thing, depending on the microscopy which you are going to perform, there are different levels of red blood cells which you require in a field of view. And for that, you are going to dilute using the buffer, and you are going to make sure that percent of red blood cells are there with respect to the total buffer which you are putting. And that's how you judge, depending on the method you are using, how much hematocrit level you are going to fix. So, after doing the centrifugation, you will get the whole blood. Taking this whole blood, we are going to further dilute it in the PBS because we have a particular hematocrit level, but that still is going to be further diluted, and we are going to actually use that particular sample for our experiments. Now, why we do this? Because first we fix the hematocrit level, and then what we are going to do is we are going to further dilute it to actually put it in the sample with respect to the PBS for our experiment. And today, not only are we going to use the PBS, we are going to change the isotonicity of the solution, and then what we are going to do is by changing the isotonicity solution, we are going to observe what is going to happen to the red blood cell.
What is isotonicity?
Isotonicity means you are trying to actually control the ionic content outside the environment for the red blood cell. Okay. Now that we are going to do using NaCl in our solution in our experiment. By taking NaCl of different amounts in the solution, we are going to see how that's going to affect the red blood cell. So these are red blood cell for different species. Like I said, not only for human beings, red blood cell is important for all the animals with blood in them. Now, these are the images which we have taken in our lab. One, this is the human red blood cell, which is of the order of 7.5 to 8 micron in size. This is mouse red blood cell, which is of the order of 6 micron in size. And this is a rabbit red blood cell, which is around 6.5 to 7.5 micron in size. Now you can actually say all of them are looking exactly the same. Then why should we even study those red blood cells? The reason is, when we are actually looking at, let us say, for example, mouse red blood cell membrane has higher cholesterol content in them. So when compared to the red blood cells of human body, cholesterol content in the red blood cell of mice is more, because of which they have more rigidity. Now when they have more rigidity, what happens? Their functionality and morphology is going to react to the forces differently. So under different conditions, to understand how the human body is going to react to the external environment, we are trying to take different blood from different animals, and we are trying to have a comparative study and try to understand how it's going to affect. Now, we can actually change the osmolarity conditions around the red blood cell, and we can try to understand what happens to a red blood cell. Now, like I was saying, when we are maintaining the red blood cells, there is a particular isotonicity which is supposed to be maintained. Osmolarity is supposed to be maintained. And why is that? Because red blood cell is actually containing the red blood cell is actually going to react to the salt solution which is outside, and we have actually listened to our previous lectures where Na plus, Ka plus and all the ions are going to actually go inside and out. Now, when there is change in the osmolarity from inside out, there is change in the concentration levels inside the cell and outside the cell. And depending on whether it is equal, whether it is greater or lesser, the liquid is always going to flow from the region of higher concentration to lower concentration. And that's exactly what is happening to the red blood cell, due to which it is going to take different shapes. Now, when we take red blood cells in a liquid of less isotonicity, it is called as hypotonic solution. And when we do that, red blood cells are going to bloat, because we are going to see normal red blood cells are disc in shape, but when you put them in hypotonic solution conditions, they are going to bloat and take the shape of the spherocytes. They are called spherocytes because they'll be spherical in shape. These are normal red blood cells, and when you increase the amount of NaCl which is there in the solution outside the red blood cells, what happens is that they are going to shrink, and these are called as echinocytes, where spicule kind of formations are going to take place for the system, because they are going to shrink, because the water content inside it is going to go out because of the change in the osmolarity. And this is what we are going to do in the lab today. What we are going to do is, all of us are going to take different concentrations of salt solution. We are going to put the red blood cell and incubate it for some time, and then we are going to see, depending on the concentration of salt in the solution which we are putting, how it is actually going to affect the red blood cell. So, how many of us are here? 1, 2, depending on the number of people who are going to do, we are going to break them into 3 people in a group, and then 3 people each can individually do a particular concentration, and they can actually look at the end product and how they are going to look. Okay, so I'll just start from one person. So continuously you change the numbers like 1, 2 and 3, so that we can group them in accordance with the number you tell. Say the number 1.
2, 3,
1. 1.
2.
Yeah, okay. You are
3, 1, 2, 3.
1, 2 and 3. Okay. All the 1s are going to take 0.6% NaCl solution. All the 2s are going to take 0.9%, and all the 3s are going to take 1.2%. Now what you are going to do is something I am going to tell you in brief. I have the whole blood solution which is already centrifuged. Now you are going to take 100 microliters of that whole blood which is diluted in the buffer saline, and then you are going to put 400 microliters of the salinity which you are assigned. Now you are in groups of 3. What you are going to do is, you are going to dilute it with respect to your saline solution. You are going to incubate it for 15 to 20 minutes, and after 15 to 20 minutes, for your salinity, how the cells are going to react is something you are going to observe in the microscope. Okay, so we will be giving you the micropipette and the micropipette tubes. Please add 100 microliter of the whole blood, the blood solution in the first, the buffer saline, 100 microliters of the blood and 400 microliters of the saline solution of your concentration. Do that and incubate it for 15 minutes.
What is this, like 12%, 1.2%, all this means, how you add? Is it
okay.
molarity, or what is the difference between osmolarity and molarity?
No, this is like weight to volume kind of a percent. If you are saying 1.2% of salt solution, you are taking 1.2 grams of NaCl and you are putting it in 100 ml of
100 ml. Okay. Not
percent. So kindly all the ones segregate in one place, two segregate in another, and please. First, all of you fill the saline in your micropipette, write your name, and then all of you can come in turns and take the whole blood and add it in there. सबको एक एक देना है. पहले वो salinity fill करेंगे depending on their number, and then everyone will put the whole.
Because it is very time sensitive. It has been already 2 things.
And note the time.
Yes.
Next.
ಇಲ್ಲಿ ಕಣ್ಣಿಂದ ತಗೊಂಡು ಬದುಕಿದೆ ಬದುಕಿದೆ.
Only English is allowed.
It's okay.
What we are doing is we are checking.
We'll see.
I think
This 3, right?
2 or 3?
3. Okay.
Give it a name. Who is doing?
So clean.
I'll clean it.
हाँ 100 microliter. Discard the tip. Second this ये line second.
नहीं.
हाँ लोग ज़्यादा है ना.
ये लाइन मेरे हाथ में 1. अभी क्या तुम लोग को ब्लड डाल.
Discard the tip there in the white.
अभी यहाँ पे लाइन में लो. पीछे जाओ.
ब्लड देना पड़ेगा.
हाँ. तो
Next. Who's left?
खुद का ब्लड नहीं खा लेते. एक ही ड्रॉप ही तो होता है.
किसी और का आप अपना दो बोलिए. तू लोग करेगा मुझे एक्स्ट्रा दे.
अरे यहाँ मेरे वो जो प्रॉब्लम आया वो पीछे.
जिसको अपना ब्लड ग्रुप
Discard
टेस्टिंग कर लो.
in the white.
नहीं वो टेस्टिंग नहीं होता.
कुछ मलेरिया
20 minutes.
नंबर
Room temperature.
अच्छा. इसमें से डालो ब्लड. स्लोली हाँ. हाँ. Slowly. Very good. Typing में करिएगा. किसी ने भी. Very good. ये मेरे सर की बात है. Thank you. हम बगल में ये डॉक्टर क्या ग्रुप नंबर तो बच्चा एक्स्ट्रा
हाँ
ये 500
इसमें ऐड कर दो राइट.
Who is this? कौन है यार? देखो ही
This is blood, right?
Yes.
Concentration.
हाँ?
Concentration.
ई पीछे किसके है?
कॉन्सेंट्रेशन इसका 5% हेमाटोक्रिट
ई
लेवल
पीछे किसके है?
अच्छा.
मतलब नाम के पीछे
नंबर
ये फर्स्ट लेटर इसका ना फेड हो गया पीई समथिंग आई डोंट नो बी और पी यहाँ किसने डाला
लेकिन बी है
ना तो ये बी है ना डी है अच्छा तुम्हारा ग्रुप क्या है वन
डिस्कार्ड द टिप there.
पता होता है ना 1.2 या 1.9
The sample was Ashmit group
मुझे मार्कर चाहिए. बिल्ड किया तूने ऐसे लिखा.
तो ग्रुप बता
3
नाम लिखा था
नहीं ऊपर मीटिंग है.
ऊपर मेंटेन ब्रॉड स्ट्रिंग
ठीक है मैं यहाँ डाल दूँगा आधा वाला एंड वाला नोट द टाइम ना
ऐसे इतना तीन कैसे
अपनी वन एक्स्ट्रा
इसने कंफ्यूज करके तीन ही बोल दिया क्या 2 या 1 का
नहीं ये दोनों लास्ट में आए थे तो मैंने
हो गया
Okay. When you are doing the pipetting, do it slowly because if the bubbles are formed, you will not be able to see properly.
normally हम directly देखते हैं ना then there is like a lot of you know inspections and all that
So these are the normal control.
we have these spacers
So you can see that they are in their discocyte shape and they retain their
you
shape.
see the sample in middle of this spacer
someone with this round
No, what happens is, unlike human red blood cell, they don't have a proper symmetry. They are like bulged on one end and small on
these
other.
are mouse
अभी तुम्हें
Yes, mice.
अभी मुझे
you can discard this
Yes, and this too. Use the paper. Discard and use this too. Bye.
कितनी है तुम्हारी 0.6 है ये तो दीदी कंक्लूजन क्या देंगे थोड़ा देख लो इसका
हाँ, कौन सा परसेंटेज है?
0.6
0.6 में they look bloated like spheres without the dimple region in between. सेंटर में अभी मैंने जब सेल्स दिखाए थे, सेंटर में डार्क दिख रहे थे और साइड्स में ब्राइट दिख रहे थे ना. Because they are like all of you have seen a donut, right? डोनट जैसे होता है रेड ब्लड सेल का शेप वैसा ही होता है. There is a dimple in the center and there is a bulge in the edge. So normal cells, which is for 0.9%, you'll actually see the dark region in the center and bulge in the outside. But for 0.6%, everything looks bright and it looks like a bloated balloon. And for 1.2%, you'll get these spicules where it's shrunk and they are crenated.
नॉर्मली
कितना है? 50. हाँ.
ह्यूमन बॉडी में क्या
क्या खून होता है?
1.9%
अच्छा ये पूछो ऑस्मोलैरिटी का पूछो. हाँ, it is comparable to the 0.9% osmolarity. हाँ? क्या हुआ? कब? अरे एक मिनट पता. अच्छा ओके. हाँ ओके ठीक. नहीं वैसे टाइम तो अभी बहुत है.
हाँ वही
कौन सा
This is 1.2. 0.6. Can you see the bloating? The center region is gone. Okay, when you are focusing out and in, you cannot see the dimple region. It's all bloated like a balloon. Okay,
एक मिनट अभी
all of them are bloated.
ये तुम्हारा 0.6 वाला ये सी इज नो उधर इट इज नॉट दैट ये सी ऐसा दिख रहा है तो थर्ड वाला है और तुम्हारा
नहीं, वो spikes क्या होता है? Once it's bloated, still the water, if it's going in, there will be a push, and due to the abnormalities on the membrane, there will be. ये डिस्कार्ड कर दो कौन
एक मिनट ये भी है
हाँ, वहीं पे वाइट पे है ना.
बस आई डोंट नो कि ये
हाँ, who's next? 1.2. हाँ, cover slip तो नहीं.
दीदी
It's on the table there.
इसकी दिखाना हाँ ये बेटर वो बेटर है
This is 1.2.
0.6.
Huh? 0.6. Okay. She said, दिगांत का 1.2 बोल रही थी तो?
ना 0.6
How much have you put?
मतलब he is saying group 3 but concentration 0.6 so 0.6 कैसे होगा 1.2 होगा ना मुझे समझ नहीं आ रहा कैसे डाल कैसे
ब्लड ना बहुत ज़्यादा डाल लिए हैं, तो उसकी वजह से ब्लड बहुत ज़्यादा डाल देते ना सैंपल में, तो क्या होता है, नॉट ओनली देयर खुद का इंट्रैक्शन बट इंट्रैक्शन विद अदर सेल्स विल क्रम्पल, तो वो हो रहा है.
100 से ज़्यादा डाल दी
बहुत ज़्यादा डाल
दिखाता
लेकिन ब्लोट तो हुए हैं, बिकॉज यू कैन सी दैट द सेंटर रीजन में कॉन्ट्रास्ट नहीं दिख रहा है, राइट? हाँ, so if you actually look at the normal red blood cell, there will be a dark region in the center because it's bent inside, biconcave shape, and in the bulge is in the edge. But here it's bloated, because of which it's like uniformly.
this was the hypotonic cell
Yeah. But he has put a lot of blood, due to which it's all
compact
Yeah.
So in normal blood
Yeah,
there
so-
is not so much, the concentration is less than this. No, not the concentration of
NaCl
NaCl, but the concentration of red blood cells.
Yes, because the blood concentration is a lot in this. Yeah. Discard this. Not the spacer. You need the space.
And this one?
Cover slip and the sample, please discard in that white. Yes, white. So
and also
this is what is the percent of this?
0.6 sir called me so I'll be waiting
Discard it. Please don't touch the table. So can you see that they're bloated? This is in reference image also I can show actually, because many people are not.
And if you go a bit further, then you look at everything else. If you want to take a certain amount of solution, you just have to go until the first point, then suck it out. And if you want to release the solution, after the first one, then the second one. Just try to get the feeling to this point.
So this is how it's supposed to look for 0.9% saline solution. And what you are getting is this. You cannot see the center, right? Because they're bloated, because of which you will not be able to.
You have to go up to the second one.
Yes, this and the carbon.
More than volume, and then it can go inside the fiber. Then to clean the fiber, it's just
Related वाले एक भी नहीं आए 1.2
No, 0.9.
0.9
1.2
Manisha.
Should I do it in one space?
Tripta, what is the spacer?
Spacer you have to keep. We have.
Where do we keep it?
Give it to Harshmeet or Suchita. Huh? Cover slips you can. Glass plates.
White spacer?
Huh?
Spacer and the
Other is
Okay.
Please don't touch the paper.
you can just touch the glass like it's not a problem.
ಇದು ಯಾವುದು
second or third?
0.9.
0.9
I'm taking the sample.
Why is it showing like this?
I think you put less blood.
I think so.
Yeah,
That's why
if
I
you
thought-
put less blood in not in the concentration which we wanted you to put, no, they'll penetrate.
Okay.
Because very less red blood cells are having a lot of salinity.
Okay.
It's created.
Yeah. Because he's put less blood in the sample.
Yeah, you can see this picture.
Yeah.
This is like a mimic of 1.2%.
That's a very
So normally it is supposed to look like this.
Okay.
certain concentration of red blood cells.
Okay. Now
But
it is less looking like this.
very less.
Because the
Yes,
salinity
okay.
is okay.
That's okay. That's fine.
Yeah, it's very strange. I have always wondered about that. Low concentrations invariably grenade.
1.2.
Finally.
ಎಷ್ಟು ಇಂಜೆಕ್ಟ್ ಮಾಡಬೇಕು?
ಅಲ್ಲಿ 50ಗೆ ಸೆಟ್ ಮಾಡಿದೆ.
ಟಾಪ್ ಇಂದ ಹೊತ್ತಗಲಿದೆ.
ಹಾಂ. ಅಲ್ಲ ಟಾಪಲ್ಲಿ ಒತ್ತಿ ತಗೋಬೇಕು. ಯು ಹ್ಯಾವ್ ಟು ಟೇಕ್ ಇಟ್ ಬೈ ಪ್ರೆಸಿಂಗ್ ಅಪ್. ಸಾರಿ? ಓಕೆ.
It's a 0.9%.
ಈ ಗ್ಲಾಸ್ ಸ್ಲೈಡಲ್ಲಿ ಹಾಕಿದ್ವಿ ತಗೊಂಡು. It's your sample. This is what's the salinity? 0.9.
No, I have to press this one.
Now inside.
While is it releasing? No, not the bubble.
It's going. Okay.
Fully. No release without bubble.
You put a lot of water. Okay.
So I've done some kind of damage to the cells.
Yeah.
This is highly possible because I played with
You
them a
have.
number of times.
Oh, okay, okay. It's okay. See, usually it's supposed to look like this.
Front is to suck
And
and then
grenaded,
front button is to suck, right?
sorry, loaded will look
Then you
like
release
this.
it. Okay, okay. Then why top?
See, this is just split.
Okay.
Loaded will look like this. Normal cells will look like this. And then when they're grenaded,
Use analog.
कि अपने सबको 20 बोले ना 20 माइक्रोलीटर लेने के सैंपल
हाँ?
20 माइक्रोलीटर
20 या 50 ऑब्जर्वेशन के लिए फर्क नहीं पड़ता है. हाँ, see, these are granulated cells for 1.2%.
Okay. Shrink one, right?
Yeah, yeah. They've just collapsed like this. Because suddenly the water from inside has gone out.
you are which group
It's got two.
two should not do
Please,
this.
No, yeah, I know that.
please discard
What
this.
did you do then
She was showing me some of the cases.
Suchita. अला दो डिस्कट
कोई बात नहीं
तेरा कौन सा?
सेंटर के हाथ में
मैं शामिल got the perfect.
No.
I think so.
1.2 अला
मैं शामिल got the perfect.
No, it already fell down. Yeah.
Very excited.
crenation.
Yeah.
Now you can see the crenation, right?
Yeah.
For 1.2. This is the reference and this is what you have got.
Take the picture, na? Manisha, take the picture.
You want?
I want. That's why. You got the perfect, huh? Oh, perfect.
Please discard.
मेरा नाम हो गया.
Yeah, who's next?
I am.
Wait, 1.2.
1.2, मतलब third. I never handled this file.
Ah, okay.
I handled simple one.
No biggie. I discarded it.
Number 3 देखना पड़ता है. Oh, sorry, for 1.2. Okay, okay.
This is just a trick I got. So you can try one. पीछे वाले
Yours is 1.2. Then
क्या हुआ?
crenation. So for 1.2 it's supposed to look like this. The spicules. And what you have gotten? Yeah, you have gotten the crenation.
Oh, then it's good.
Please discard this. What is the salinity for your sample? 1.2.
इसका क्या करूँ?
हो गया? आपने विजुअलाइज कर लिया?
नहीं.
अच्छा वेट कहीं कहाँ कहीं पे भी कर सकते हैं. नॉर्मल सेल दिखाती हूँ. यहाँ पे रेफरेंस रखा है. This is 1.2, right?
क्योंकि हमारा एकदम गोल गोल
Okay. So you can see the spicules here. It's like crumbled like this. So this is how it's supposed to look, and that's how you got.
And if excess of NaCl is in our body, so it's burst, RBC burst.
Yeah. The beauty of our body is it's maintained so meticulously that it doesn't happen. Thank God that it doesn't happen.
But during
But kidney takes a hit.
हाई ब्लड प्रेशर में बोलते हैं NaCl नहीं दे मतलब
Kidney takes a hit. वहाँ पे क्या होता है? See, high blood pressure is not only related to the salinity. It is related to so many other factors. ठीक है? If you have diabetes, if you have cardiovascular issues, if you have bone marrow issues, there are many reasons due to which it becomes more and more rigid, due to which it's difficult to pass through the blood vessels and pressure बढ़ जाता है ना? Force per area बढ़ जाता है. That is your blood pressure.
during accident when we lose lot of blood, sometimes
It is
accident
almost like using a conflict.
This is not confidence.
This is so yeah, it is contrast,
Yeah.
but the user interface in software
Yeah. This is size
because when I
and inverted. No? even in that you use inert.
Right, right. When I was generally not focused.
Yours is 1.2?
ब्लड इमीडिएटली लूज होगा तो तुम्हारा
मेरा
आयरन
3 नंबर था.
Ah, 1.2. Okay. Put in a lot of blood cells. Okay. But then, like, you can see that they're crenated, right?
Right. Achha, they have like.
Suppose it will look like this. Crenated cells look like this. And what you have gotten is this.
Can I take a picture?
Wait, I'll see if I can get a better view of yours.
You are group 3.
Group 2.
Group 3, either 1 or 3.
अरे वो मार्कर से
अच्छा, हाँ.
हमारा
तुम्हारा 1 था, तुम्हारा 3. That's 2 3 is hypertonic.
हाँ. Hypertonic. Right. इसलिए तो सिकुड़ गया है.
हाँ, इसीलिए वो तुम्हारा वो solution
I don't know whether you'll be able to.
निकल गया है.
I don't know whether you'll be able to make out the shape change here. ऐसे दिख रहे हैं लेकिन बहुत सारे हैं ना तो.
Okay. तो मैं zoom हो कैसा रहा?
इसमें 2 ड्रॉप्स है.
ये देख के लग रहा है कि इसमें बहुत ज्यादा ब्लड है.
हाँ बहुत ज़्यादा डाल दिया है आपने.
You can look, मतलब तुम्हें दोबारा देखना है कंप्लीट डाल के. Do you still have time ना?
उनका ब्लड डाल के भी देख सकते
ये सैंपल में ये प्रॉपर्टी.
ये क्यों दो है
एक हमसे गलत मतलब इरेगुलर शेप.
बट सेम लिक्विड है ना.
Same.
No, that doesn't matter.
No, that one.
I usually do this to save time.
This is my yeah. This one's fine.
मतलब if I want to observe two things just for checking.
कलर देख के पहचान लिया.
कलर अच्छा तो बाकी को What is this H? पूरा नाम लिखा होगा.
See, मैंने बिना देखे पहचान लिया कि नहीं.
Blood red color के को सबके साथ जड़ी.
नहीं, it's not about it. It's about how dark.
Seriously.
Just compare this and this. आप बताओ दीदी कौन सा ज्यादा है?
ज़्यादा ब्लड डाल दिया।
ज़्यादा ब्लड डाल दिया.
उसका 1.2 भी कितना?
मार्केट चल रहा है ना आपने देखा होगा.
आपका कितना था?
1 ग्रुप 1 था.
हाँ?
ग्रुप 1 था.
Group 1.6 ना?
Yes.
हाँ, okay. So you know that when we speak about red blood cells, they have V shape, na?
Yes, ma'am.
Discocyte shape, where in the center you have a dip and a bulge, right? But you have 0.6% in yours, so they're supposed to bloat like this, and that's why they're bloated without the dimple. So you've put in the right.
तो अगर हम 0.6 को ही बहुत ज़्यादा इंक्यूबेशन टाइम में रखेंगे तो डज इट अफेक्ट द शेप?
Of course it will, क्योंकि वो gradually use होता है ना. जो भी salinity में आप डाल रहे हो, it is continuously being maintained. And how long will it maintain? Till the previous buffer cell and everything is maintained, and osmolarity is maintained. वो तो gradually खत्म ही होता है ना, because red blood cell is continuously using it. आपका कितना है salinity?
1.2. क्या करा था मैं? थोड़ा सा आउटसाइड पेरिफेरी में देखने से आई थिंक
हाँ.
वो नहीं दूसरा.
आपका कितना था?
वो
1.2.
1.2.
तो आपका क्रम्बल होके क्रेनेट हुआ है,
ओके.
which is how it's supposed to look like.
मैं फोटो खींच रहा हूँ.
हाँ आपका खींचना, ये रेफरेंस है और ये आप।
इतना भी एक ये भी सैंपल है, इन लोगों को पूछो छोटे उसका सैंपल।
अच्छा हाँ, उसका भी 1.2 था ना।
लेट देम गेस, लेट देम गेस।
हाँ ओके,
सबको बुला के पूछे
यहीं पे एग्जाम हो जाएगा।
सबको तो ऐसे सुबह जो मैंने लिया था ना वो रेफरेंस दिखा के ही बता रही हूँ, तो होपफुली
तो ये एकदम थोड़ा वैसे लग रहा है, न्यूट्रोफिल से इजोनोफिल में जो वो जैसे होते हैं उसकी तरह शेप्स आ रहे हैं। हॉर्सशू शेप,
हॉर्सशू नहीं, समथिंग क्या हॉर्सशू शेप को कोई न्यूक्लियस होता है हमने एक भी करा दी
Oh yeah.
वही पेपर का थोड़ा सा है।
वो सही से नहीं, सही से नहीं।